It's a real pleasure to talk to you today with my colleague, Professor Hart. This is a subject matter that's close to both of our hearts and something we've both been scratching our heads over for probably the last 20 years, seeing patients in the clinic and also doing research in the laboratory. So I'm going to just give some introduction to inflammatory bowel disease to start with, and then also talk about a problem that's a common one for us, and that's that a lot of the drugs that we're using, even though we've got a vast armamentarium of them now, many of them very expensive, don't pretend to work so well in all of our patients, and that's a real problem for us and for our patients. So that's something I want to hopefully drill into. So what is inflammatory bowel or IBD, inflammatory bowel disease, or IBD as we often call it? So this is what a healthy gut looks like. You can see that it starts with the mouth, goes down to the food pipe, into the stomach, lots of small bowel, and then into the large bowel. And just some interesting facts about that normal GI tract. So it's about 25 feet long, maybe 30 feet in some people. And if you unravel it out, particularly with the little villi that are sort of pointing out, the surface area of it is about the size of a tennis court. So pretty big surface area exposed to the external world. We call it the second brain as well. There's over 100 billion nerves in the gut, which is close rival to the brain, which tells you that this is an organ that needs a lot of control. And it's also an extremely hostile environment. There's over 40 billion bacteria, trillion bacteria, I should say, that occupy that niche. And there's viruses, there's fungi, and of course, lots of food trafficking through there every day. So it's a particularly hostile environment, which is why I think some of us are prone to developing this problem. So inflammatory bowel disease is two diseases really, or two main diseases that we call ulcerative colitis and Crohn's disease. Ulcerative colitis, you can see, so these are our endoscopy pictures that you can see here. So when we stick a telescope through the back passage, advance it into the colon, where we can visualize it and take samples from it. In the healthy situation, you see the healthy patient looks nice and pink. You can see lots of nice blood vessels. In the ulcerative colitis patient, you can see it's red, it's angry, it's bleeding, and there's ulcers. And it's a very similar situation with Crohn's disease. Ulcerative colitis tends to affect just the colon. You can see it's red in just the colon here, whereas Crohn's disease can affect anywhere along that GI tract. And actually, it also tends to come back even if you do surgery on it. This is a patient of mine who's had an operation to try to cut out that bit of Crohn's disease affecting the small bowel. This is an endoscopy done 6 months afterwards, showing that it's recurred, a big ulcer where it's been joined up. So this is a disease, if you treat it with surgery, tends to come back. But I'll talk more about surgery in a moment. So what problems does it cause for our patients? Well, it won't surprise you when you've got a very inflamed bowel that some of the symptoms our patients experience is torrential diarrhea, which can be accompanied by bleeding. So rectal bleeding can cause quite significant abdominal pain, and patients often experience loss of appetite. Fatigue is also really common in our patients. And in fact, there's a significant burden of psychological morbidity in our patients, anxiety and depression associated with these really quite embarrassing sometimes symptoms and debilitating symptoms. There's lots of other manifestations that it's associated with as well. Patients get inflammation in their eye. You can see that little collection of pus collected underneath the iris there. There are also increased risk of developing cancer. You can see this lesion again with a telescope test here, where a patient has developed colorectal cancer. A fistula is probably an unusual term to many, but it's something we're very familiar with in our patient group. So patients with Crohn's disease develop these abnormal connections in the back passage where it's inflamed, connecting the anus or the rectum to the skin outside, and you can have a discharge of pus, and it can be very painful and get infected and causes lots of problems for patients. We also see inflammation of the joints. And you can see this left knee here is very swollen. It would be painful because of an arthritis in the knee. And we also see inflammation in the skin. So these painful nodules here called erythema nodosum, and this nasty ulcerating lesion that we see in the shin here called pyoderma gangrenosum. And it's not just our patients that really get the sharp end of this. This causes a significant burden on society as well, in the sense that in fact, this is an underestimate that I'm giving you now. It's more like approaching 1% of our population. So 1 in 100 of our patients are now, of our communities, of our population of the UK, are experience IBD. And what I mean by burden, I mean that it costs us all money because the healthcare consumption associated with IBD is now approaching £1 billion per year. And the lion's share of that cost is the expensive drugs that we use, which is why getting treatment right and getting effective treatments is so important. And it's also now a global disease. So it was very much a disease of the West in years gone by, but almost unheard of in the developing world. But we're now seeing burgeoning cases, particularly as the developing world industrializes around the world. So India in particular, there's probably 1.4 million cases of IBD in India now as it sort of industrializes. Why do people get it? Well, we don't really know is the answer, but there's a whole bunch of reasons that are speculated. We know that genetic susceptibility is a thing. It's not a single gene, like something like cystic fibrosis, where if you've got an abnormal gene, you get the disease. There's a whole bunch of genes that confer a small amount of risk. And in fact, I'd argue IBD is probably the most complex genetic disease now known to man. There's over 400 genes that have been linked to associating risk. But we know it's not all genes. So if you've got an identical twin, for instance, who've got exactly the same genes as their twin. So if you've got Crohn's disease in one twin, then the risk of the other one having Crohn's disease is probably about 40%. So clearly there is a genetic component, but there's more to it than genes. And increasingly, we recognize environmental features as being really important, especially our diet. Increasingly, the kind of composition or the community profile of bugs that live in our gut. That 40 trillion we've co-evolved with over many years, but we know in IBD the profile of those gut bugs has changed. And there's also other things, not just our diet, but also things like smoking, where we live in the world, influences our susceptibility. And what we also know is our immune system, which of course has evolved to help defend us from pathogens and fight cancer. If that becomes inappropriately activated, we can get these immune-mediated inflammatory diseases. And IBD is one of those, just like arthritis, psoriasis, multiple sclerosis, where the immune system is inappropriately activated against something that causes these problems. Who gets it? Well, we're all at risk of it. Here's some people that many of you will recognize. These are all people who have told us that they have IBD. It's a disease often affects young people in the 20s to 40s, but also there's a second peak as people get older, and it's created some interesting issues. So Dwight Eisenhower had Crohn's disease, and in fact, when he was anesthetized to have that taken out, it did raise questions about executive power when the president is incapacitated. And of course, that came to pass when JFK was assassinated, and how executive power passed on to the beyond the president. Amy, you'll recognize from Strictly Come Dancing. She's got Crohn's disease. And a bunch of sportsmen as well. This, as I said, this disease often affects young people. Lewis Moody, I hope you recognise as the man who won the line-out before we won the World Cup in 2003. He had to move close to Twickenham because he had such bad ulcerative colitis. He was so worried about incontinence and problems like that he had to move where he actually lived close to his workplace. Jack Leach has missed many games of cricket and many tours for England because of Crohn's disease. And unfortunately, Shinzo Abe, who retired, unfortunately was assassinated a year or two back, but had to retire from the presidency of Japan because of really bad ulcerative colitis. So how do we treat it? Well, I'm not going to go through all of these treatments, but just to say there is a vast array of them now, including a lot of tablets that we use and some more advanced therapies, which are essentially antibodies that have been bioengineered to specifically target some aspects of the immune system. Even then, 50% of patients with Crohn's disease will need surgery, and probably 20% of patients with ulcerative colitis. So even despite all these great advances, and they are advances, we still need to operate on quite a few of our patients. And how effective are they? And this is the nub of what I wanted to get to, in a sense, in that even these are our most commonly used drugs that we use to treat ulcerative colitis, for instance. So infliximab is an anti-TNF antibody. Vedolizumab is another biologic antibody that stops immune cells from trafficking to the gut. And ustekinumab is another antibody that blocks another cytokine. And these are our key clinical trials that got these drugs approved for ulcerative colitis. And you can see after 1 year of treatment, how many of our patients have got themselves fully into remission, are better and back and functioning as normal. Well, it's a sorry story in some ways, in that you can see the treatment group here, how many of them are achieving that endpoint of clinical remission. It's way fewer than half of our patients after 1 year of treatment. So if you look at it in terms of these 100 patients here, the green patients have got into remission, whereas the red ones have still got active disease. So you can see that treatment resistance is really a big problem for our patients. So some of the research that we've been doing at Imperial is to try to address this problem. Why are people resistant to treatment? And what are the mechanisms of that? And if we understand that, can we bypass that resistance pathway and get better treatments? Or can we understand the molecules that are contributing to resistance? And that way, at least we could potentially identify our patients who might be resistant, so we could take alternative therapeutic strategies with them. So this is the kind of approach that we've been taking in the lab. So we have a group of patients with inflammatory bowel disease who are about to undergo treatment, and then we follow them up, and then we can determine whether they responded or not responded to treatment. And then one of the things that we can do is at the beginning, before they've had treatment, we can take lots of samples, so biopsies from the colon when we do our endoscopies, we can take samples of stool and of course blood tests. And what we can do, we can look for things that are present in those samples in the patients before they've been treated. And because we know their outcomes, we can try and map that to whether there were any features that were predictive of people becoming responders, or indeed that predicted resistance. And there's a whole host of clever stuff that we can do now that based on the patient's genetic profile, what genes are switched on and off, and the type of bugs that are colonizing their gut, the kind of proteins that are being produced. So lots of what we call multi-omic technologies to try to look at that group of patients before they've been treated to see if there were predictors of how they subsequently then responded to treatment. So here's one of the experiments that we've done. So a big group of ulcerative colitis patients, so over 300 of them, who were treated with one of these drugs that I mentioned to you earlier, ustekinumab, and then we defined them as responders or non-responders to treatment. A biopsy was taken from the colonoscopy before they had treatment. And what we were able to do with that, we were able to look at what genes in that tissue were switched on and which ones were switched off. So which ones were activated, which ones were not. And you can do this kind of experiment. You can perform these kind of arrays where, remember, there's probably 25,000 genes in a human. So across that genome, you can see which genes have been activated and switched on and which ones are not. And this is what we hope to see. So a different pattern of genes are switched on in responders and a different pattern in non-responders. And if we can build up that kind of profile, then we can try to understand what's causing response and resistance. This is quite a complicated slide, but I'll boil it down to some simple things. So what we did is, in the non-responders, we looked at which genes were activated and found some interesting patterns. A group of genes that are co-expressed together, we call a module, because they always share the same expression pattern. And we found at least 3 modules that contain genes that was really strongly associated with treatment resistance in this big cohort of patients. I'm not going to go through the individual genes, but you can see these have been sort of displayed as a network because these are often genes, as I said, are co-expressed, and they often share a function, which was what we were very interested in here, because as well as identifying these networks, we're very keen on understanding what are the biological processes, what are the mechanisms by which these genes are working together to drive resistance in our patients. So this is what we call a pathway analysis. So based on those patterns of genes that we see up or down, so activated or inhibited, we can identify specific pathways that are linked to treatment resistance. And if we summarize some of those things, there's an immune cell, which is essentially the infantry of the immune system that we call the neutrophil. There's many of them in the body, and we find them trafficking into the gut in patients with colitis. And what we found is those patients who are associated with treatment resistance are highly enriched for these neutrophils, and they've got lots of granules in them, and when they release that cargo by degranulating, there's lots of toxic molecules in there, and that was strongly associated with treatment resistance. And likewise, some of the proteins or the molecules are responsible for mediating the signals of the immune system, or in fact you could even call it the language of the immune system, are cytokines. So we also found that was a key pathway. And also we've mentioned the importance of microbes in our gut. We also found pathways that were linked to how our immune system perceives, recognizes, senses bacteria in the gut were linked to treatment resistance. One of the other modules, TM16, that was associated with what we call extracellular matrix dysregulation. And what does that mouthful mean? Well, what that means is our tissue, as well as having lots of immune cells in it and the sort of cells that are required for it to form its function, they often have really important scaffold cells there that give you the sort of scaffolding for you to build your tissue on. And that was really significantly dysregulated in patients who were resistant to treatment. We also found some aspects of how we synthesize our proteins and something called ER stress, which is again the apparatus in our cells that makes protein look to be stressed in patients who were resistant to treatment. And remember, this was a drug called ustekinumab, so we were very interested whether this is actually a pattern that we see in patients treated with other drugs. So we looked in another 2 independent studies to see if we're seeing the same problem in patients who are also resistant to another drug that we use to treat IBD, and that's called anti-TNF. So again, you know the drill. We take the biopsy from the patients before they've had their treatment. We check which genes are up and which ones are down and do our assays. And again, it's quite a complicated graph, but what this is showing you here now is if that pathway that we knew was associated with resistant is enriched, it goes up the axis here. If it's not, it goes down the axis. And in study 1, we've divided our patients again, like before, into responders and non-responders to treatment. So if you're in claret, you're a non-responder to treatment. If you're in blue, you are a responder to treatment. And you could see that the way in which those modules were expressed was completely different in those patients who responded. The clarets were all enriched for the module we'd seen before, whereas the blues had very low level of that module, exactly as we'd seen with ustekinumab. And in a second independent study, we saw exactly the same thing. Those patterns of resistance, molecular resistance, are appearing in those exact same patients. Remember, this is before they've been treated. So this is a signal we can detect in advance of a patient being treated. I'm not going to talk about this slide in any great detail, but other than to say what we can do with some of these data, because we know how individual cells express some of these genes, we can go back into the gut and look at single cell level and find out which cells are expressing those genes, which cells those ones are talking to, and we're able to build these complex interactive maps that telling us, based on the genes that are going up or down, which cells are talking to each other. And there's lots of immune cells involved in this, and some of those scaffolding cells that I mentioned, what we call stromal cells. We're now confirming these findings in a new cohort of patients, because if we can confirm that this signature that we've detected can predict resistance to treatment, that could change how we think about treating our patients. And this is a study we started over a year ago now, and we've already recruited over 100 patients who are all starting new treatments. And again, you know the drill. We're going to take that biopsy from the patient at endoscopy before they get treated. We'll also look at the kind of bugs. I've not had time in this talk to talk to you about how the bugs might change and predict treatments, and likewise some of the features that we see in blood, like protein, or even the genes expressed in blood, again, how they might predict treatment. So in conclusion, hopefully you share my view that IBD is not just a common disease, but it can be a severe disease that can really impact on the lives and the quality of lives of our patients. We're very fortunate that we now have lots of treatments to use for our patients, but the downside to that is that fewer than half of them manage to maintain sustainable, durable, long-term remission, which is, of course, the goal that both our patients and the clinicians that treat them aim for. I think there is room for hope here, that we're now gaining enough agility around understanding and profiling the kind of molecules that are present in patients at the beginning of their journey to try to get some insights into what is causing patients to be resistant to their treatments. And there are really complex crosstalks and dialogues and circuitries between some of the important immune cells and scaffolding cells that live in the gut that are probably driving this process. And understanding that process might indeed give us treatments that are not prone to this resistance problem if we really understand that circuitry in greater detail. And I also think that this type of approach of molecular profiling is, and what we're trying to do with this, the latter study that I mentioned, the TEM study, is really to develop a biomarker-based platform where we can predict patients who are resistant to treatment at the beginning of their journey, so that we've got other options for thinking about how we might treat those patients. And perhaps that might be something for discussion. And then finally, I did just want to acknowledge the big group of research people that goes beyond this enormous amount of work. These are the sort of the stars that make this all happen. So I really did want to pay tribute to those, and of course the research funding bodies that support this work, which of course none of this would be possible without as well. So that's it from me.